Hello, and welcome to the Clinical Care Options Oncology Podcast. I'm your host, Tim Quill. Today's episode features Dr. Kathleen Moore from the University of Oklahoma in Oklahoma City and Dr. Nicoletta Colombo from the University Milan Bicocca in Milan. They will be discussing key updates and new clinical trial data for cervical, ovarian, and endometrial cancers that were presented at the 2023 ASCO Annual Meeting. This episode is part of a larger educational program entitled Key Findings and Expert Recommendations in Gynecologic Malignancies, Independent 2023 Conference Congress Coverage. For more information on Dr. Moore and Dr. Colombo, along with a link to the complete program, including a downloadable slide set and clinical commentaries, please visit the show notes for this episode. Now let's get started and hear what the experts have to say about key data presented at the 2023 ASCO Annual Meeting.
My name is Dr. Kathleen Moore, and I am Associate Director of Clinical Research and Professor in the section of Gynecologic Oncology at the Stevenson Cancer Center in Oklahoma City, Oklahoma. It is my great pleasure to be joined today by Dr. Nicoletta Colombo, who is an Associate Professor of Obstetrics and Gynecology at the University of Milan in Milan, Italy. Welcome to Dr. Colombo, and welcome to our listeners. Today, we will be highlighting for our listeners some of the key studies that were presented at the 2023 American Society of Clinical Oncology Annual Meeting held in Chicago, Illinois. We will be focusing on key data presented for endometrial and ovarian cancer with the potential to change the standard of care, final overall survival data from KEYNOTE-826, and a trial from the Canadian Cancer Trials Group comparing radical hysterectomy and pelvic node dissection versus simple hysterectomy and pelvic node dissection in patients with low-risk early-stage cervical cancer, which is called the SHAPE trial. First, I would like to start with key studies in ovarian cancer, including data from the confirmatory MIRASOL trial, and then the one that I alluded to, the Canadian Cancer Trials Group. So let's start with the MIRASOL study. MIRASOL is a randomized phase 3 trial that compared the antibody-drug conjugate mirvetuximab soravtansine to investigator's choice chemotherapy in the platinum-resistant recurrent status tumors that are folate receptor alpha high. Patients could be enrolled on the MIRASOL study if they, again, had platinum-resistant disease. They could have 1 to 3 prior lines of chemotherapy, folate receptor alpha high. They did not have to have bevacizumab, but they could have had bevacizumab, which is in contradistinction to the SORAYA study, which is what garnered accelerated approval in the United States. And they did not have to have a PARP inhibitor unless they had a BRCA mutation, but they could. And I'll give the breakdown of demographics in a moment. Patients were then randomized 1-to-1 to mirvetuximab, 6 milligrams per kilogram IV every 3 weeks until progression or toxicity, as compared to investigator's choice chemotherapy, which could be weekly paclitaxel or pegylated-liposomal doxorubicin, which I'll call Doxil from now on, or topotecan. And the primary endpoint was progression-free survival, measured by an investigator. And the key secondary analytic endpoints were overall response rate and overall survival, and as well as patient-reported outcomes. So at ASCO, we heard the primary endpoint of progression-free survival, as well as overall response rate and overall survival. So this study enrolled 453 patients. As I alluded to, they did not have to have prior bevacizumab, but 60% of them, a little more than that, did when they came on study, and about 55% of them had a prior PARP inhibitor, which is consistent with current global availability. In terms of the known stratification factors, or the planned stratification factors for the trial, which were number of prior lines of chemo and then investigator's choice selection, the majority of patients came on with 3 prior lines of chemo. So like 46% came on with 3 prior lines of chemo, only 14% with 1 prior line, and then the rest had 2 prior lines. Weekly paclitaxel was the most commonly selected investigator's choice chemotherapy at 41%, a little less than that for Doxil at 36%, and then 23% elected for topotecan. The primary endpoint was progression-free survival, and this endpoint was met with a hazard ratio of 0.65. So that is a 35% reduction in the hazard of progression with use of mirvetuximab as compared to investigator's choice chemotherapy with a p-value of 0.0001. And I would encourage you to look at the link so you can look at the survival curve, because importantly, what you see in the control arm is what we see in every negative phase 3 study, which is basically everything we have done since Aurelia. About 50% of patients who participate in these trials, irrespective of the arm, discontinue at the first disease assessment. And that's what you see in the control arm here. At that 6-week mark, 50% are already off. We do not see that with mirvetuximab. There's an early separation of the curves that is maintained, and we do not see this 50% drop off. In fact, at the first disease assessment, we still have almost 90% of patients on mirvetuximab. Because the primary endpoint was met, we could roll alpha to the key secondary analytic endpoints, which include overall response rate. And here, mirvetuximab exceeded investigator's choice chemotherapy, 42% overall response rate versus 16%. And that 16% is exactly what was included in the statistical assumptions of the trial for the control group. Notably, there was a 5% complete response rate with mirvetuximab and 0 for investigator's choice. And this was statistically significant with an odds ratio of 3.8 and a p-value less than 0.0001. So because that was positive, we were able to shift all of the alpha to the interim overall survival analysis that was pre-planned at the time of the progression-free survival readout. And statistically, at this point, for overall survival to be read as positive, the p-value would have to be less than 0.0131. And so what we found with overall survival, with 13.1 months of follow-up and 68% of final overall survival achieved, the hazard ratio was 0.67. So a 33% reduction in the hazard of death with use of mirvetuximab as compared to chemotherapy, with a p-value that was less than that pre-specified boundary. Here it was 0.0046. So this is statistically and clinically significant improvement in overall survival. From a safety standpoint, we did not see any new adverse events from those previously described in prior clinical trials, including the single-arm phase 3 study SORAYA. And so mirvetuximab is characterized by common but low severity ocular and GI events for which preventative and mitigation strategies are in place and work well. And in fact, only 4 patients discontinued the mirvetuximab arm due to ocular toxicities. So in summary, for the MIRASOL study, this is the first study ever of a novel agent in a phase 3 trial to demonstrate an overall survival benefit in the platinum-resistant ovarian cancer space. This is the first antibody-drug conjugate approved in ovarian cancer, and this is the first biomarker-directed therapy available in the platinum-resistant setting, and only the second biomarker-directed therapy available in ovarian cancer after PARP inhibitors. So a lot of achievements here. This is already available in the United States based on accelerated approval from this SORAYA study. This is anticipated to confirm that approval, but perhaps more importantly, will allow for global authorization of this medication, and so patients outside the US can access it. The second study I'm going to discuss is a very exciting study and perhaps one of the top 10 most important studies presented at ASCO, and this is the SHAPE trial, which is an international randomized phase 3 trial comparing radical hysterectomy and pelvic node dissection versus simple hysterectomy and pelvic node dissection, importantly, in patients with low-risk early-stage cervical cancer. And as this audience likely knows, the standard of care for early-stage cervical cancer has been radical hysterectomy for decades. And we all know that the quality of this surgery has been incredibly important. We get one opportunity to cure cervical cancer. And if you don't, when these tumors recur, especially in this day and age, they are largely incurable. The trial schema for the SHAPE trial was a randomized phase 3 study that randomized women with early stage cervical cancer to one of 2 arms. One, which is radical hysterectomy, and the second is a simple hysterectomy. Now, there are a couple important points to make here. Number one is, regardless of the assignment of the surgical technique, both types of hysterectomy required that a pelvic lymph node dissection be performed. And surgeons had the option of doing a sentinel lymph node mapping if they chose to, but lymph nodes were required here The patients who are included in this clinical trial were highly selected to be appropriate for considering this simple hysterectomy approach. Early small studies have really shown that in these smaller tumors, when they look at the pathology evaluation, there is no spread to the parametria. It's just not where they go. They can go to lymph nodes, but they're not in the parametria, and they're not in these like extensive vaginal resection margins, which really justified asking this question. But it had to be carefully selected. So these were squamous cell or adenocarcinomas or adenosquamous. So not your neuroendocrine or any of the rare. These are kind of standard squamous or adenos. Stage 1A2 and 1B1 tumors with less than 10 millimeters of stromal invasion on a leaf or cone specimen. They had to have less than 50% stromal invasion on an MRI, and they had to have a smaller tumor than 2 centimeters or 20 millimeter. So these were small tumors, really looking at the kind of tumors that you ideally detect through pap smears, right? That's who is included in this de-escalation trial. Primary endpoint was pelvic recurrence at 3 years. Why is that the endpoint? This is the endpoint because most recurrences in following radical hysterectomy for cervical cancer really happen within the first 2 years, and they extended to 3 just to be conservative. And so pelvic recurrence rate was really the endpoint here. We're not looking for distant failures. What impact did the type of surgery have on the pelvic recurrence rate? And then, of course, they're looking at everything else: pelvic relapse-free survival, extra pelvic relapse-free survival, so the distant disease, overall survival, etc. So very well designed study, and this was a non-inferiority trial. And so so these are different than what we're used to seeing with superiority trials. What are the statistical kind of boundaries for saying this is non-inferior? How do you say something's non-inferior? So non-inferiority of simple hist to radical hist could be claimed, this was the statistical assumption, when the upper one-sided 95% confidence interval for the difference in the pelvic recurrence rate for simple to radical hist had to be less than 4%. 700 patients were randomized, and it took 7 years, 2012 to 2019, to get this accrued. And this is really a testament to Dr. Plant and her team's commitment to getting this study done, and also to the patients who participated. This is a hard thing to randomize to. I'm going to randomize you to standard surgery, which I know works really well and has worked for decades, versus a new, maybe less morbid procedure, but maybe it won't work as well. It's a big ask of our patients. So impressive. We had equal distribution, 350 patients in each arm, simple hysterectomy versus radical hysterectomy. Numerically, a little bit more patients in the simple hysterectomy group had a leap cone preceding their procedure, so 73 versus 65%. So that's a little bit of an imbalance, but not really that great. Most patients came on with 1B1 disease, about 92% in each arm. Most patients came on with squamous cell disease, about 62% in each arm. And most were grade 1 to 2, which is right about 60%, which is about what we see. So it represents, the tumors that we take care of. Now, this study did not say it all had to be done open. Providers could decide if they wanted to do a minimally invasive or an open approach. So in the simple hysterectomy arm, 17% were done abdominally and 56% were done laparoscopically. An additional 24% were done robotically and vaginally. So the take home is 84% were done not through an open procedure. Whereas if you look at the radical hysterectomy arm, 29% were done abdominally, and the remainder, so about 70%, were done minimally invasively. So that's a little bit different. Sentinel node mapping was planned equally in both arms at about 37%. So the take-home kind of messages are the following. 1, for pelvic recurrences, simple hysterectomy group, 3.1%. Radical hysterectomy group, 2.9%. So we had a 0.2% higher risk of pelvic recurrences in simple versus radical hysterectomy, which is clearly non-inferior. Extra pelvic recurrences, 2% in the simple hysterectomy group, 0.6% in the radical hysterectomy group, for a cumulative extra pelvic recurrence risk of 1.3%. Deaths due to cervical cancer, 1.1% in the simple hysterectomy arm, 0.3% in the radical hysterectomy arm. When we look at the pelvic recurrence rate in the intention to treat population, the pelvic recurrence rate at 3 years for simple hysterectomy was 2.52%, and for radical hysterectomy was 2.17%. The difference is 0.35%, with the upper 95% confidence limit of 2.32%. And remember, I told you that boundary had to be 4% or less. So it clearly met that bar. So we can say non-inferiority of simple hysterectomy to radical hysterectomy could be concluded. There were no subgroups that disputed that. If they looked at tumor grade, histologic type, histologic stage, etc., all favored the simple hysterectomy randomization or selection as per the primary endpoint. They did look at overall survival, 99.1 versus 99.4%, so clearly no difference. So adverse events were interesting. We think that radical hysterectomy is a more invasive procedure, and indeed it is. Any adverse event within 4 weeks of surgery, 42% simple hyst versus 50% in radical hysterectomy. After 4 weeks, so like late recurrences, 54 versus 61%. So it wasn't statistically significant, but was numerically trending for late with a p-value of 0.08. But the early complications were statistically significant with a p-value of 0.04. Really saw the difference was in urinary complications. There were statistically significantly more urinary incontinence or urinary retention for early complications for radical versus simple hysterectomy, as well as late complications. And I think this is important. 11%, this is after 4 weeks of surgery, so the retained catheter is out. 11% urinary incontinence versus only 4.7% in the simple hysterectomy group, as in the same trend for urinary retention, 9.9%, which is what you expect with the rad hyst, versus basically none for urinary retention. Both of those highly statistically significant. And so the take home from this really is in early stage low risk cervical cancer, pelvic recurrence rate as 3 years with simple hysterectomy was not inferior to radical hysterectomy. And there were fewer urologic surgical complications seen following simple hyst. And I didn't talk about it, but please look at the presentation. They also did quality of life and sexual health measures, which were better seen, following simple hysterectomy as compared to radical hysterectomy. So the most important take home, and Dr. Plant made this in her presentation, is following adequate and rigorous preoperative assessment key. Adequate and rigorous. These decisions have to be very carefully made. But if you've done that, simple hysterectomy can now be considered the new standard of care for patients with low risk early stage cervical cancer. Again, stage 1A2 to 1B1, less than 2 centimeters, less than 10 millimeters of invasion on a LEAP, or less than 50% depth of invasion on MRI. That's the consideration. So for those patients, the standard of care, once we see the manuscript, will likely shift.
Dr. Moore, the majority of the patients in this trial underwent minimally invasive surgery, and there was actually a slight imbalance with more patients undergoing laparoscopy in the simple hysterectomy group. How do you interpret these results in the light of the LAC trial? Does this mean that laparoscopic simple hysterectomy is safe, but laparoscopic radical hysterectomy is not for cervical cancer? And again, in this respect, do you believe that the higher proportion of extrapelvic recurrences in the simple hysterectomy group may be related to the laparoscopic approach?
Thank you, Dr. Colombo. This is one of the most important questions following the SHAPE trial and really following the LAC trial, which we all know, and really the LAC trial really put an end to minimally invasive, primarily laparoscopic radical hysterectomies globally. And we all switched back to doing open procedures, at least in the United States, because that particular study of minimally invasive versus open radical hysterectomy demonstrated a higher risk of death with the minimally invasive approach, which was largely laparoscopic in that trial. There was very little robotic use. And so that really changed operative approach in the US at least. And here we see the SHAPE trial, which has been going on for a long time and really overlapped with LAC. We have to acknowledge that. where we do see a very large percentage of patients who were treated with a minimally invasive approach. Here we actually see more use of robotic interventions, and about a quarter of patients in each arm had a robotic intervention, and the remainder, in the upper 40s to 50% were laparoscopic. So cumulatively, you see this minimally invasive approach. I think that what we can say from the SHAPE trial is that, one, the surgical approach, other than simple versus radical, was not an analytic endpoint. And so we cannot conclude from the SHAPE trial that minimally invasive approaches can come back as an approach for radical, and I would even say for simple hysterectomy in the setting of a cervical cancer. I do not think we can say that at this point just yet. I do think this gives us a sense that it's probably, in this population of small tumors, safer than what we saw in the LAC trial, where there were some larger tumors included there. The hypothesis from the LAC trial was, of course, the surprisingly high rate of recurrences was because probably several things, but the main working hypothesis comes down to tumor containment. And so here, which is kind of, again, this predates LAC or overlaps LAC quite a bit, there was none of that. People were just doing radical hysterectomies and simple hysterectomies, minimally invasive in the way that they knew how, probably laparoscopic assisted vaginal hysterectomies or total laparoscopic hys without containment mechanisms. And so it is possible that this little blip up, it's not big, but you do see a numerical difference here in the extrapelvic recurrences. Could that be because of that lack of tumor containment? And so I think that this is an ongoing question. And I would be very cautious to discard the LAC findings in this population and just adopt minimally invasive as the new standard of care, especially if you're not doing containment procedures. The ongoing ROC trial that is led by Dr. Kristen Bixel and Dr. Mario Lateo is looking at radical hysterectomy done minimally invasive robotically versus open. And it will have some of these smaller tumors in it. Honestly, I think continued enrollment to that study with potentially some amendments is really justified to make sure that we are really sure that we're not leaving anyone behind and increasing the risk of recurrence with this minimally invasive procedure and or need for containment procedures. So more to come on this question, but that is really the elephant in the room for what we need to figure out moving forward.
In the Aurelia study, the addition of bevacizumab to chemotherapy, particularly paclitaxel, provided a great benefit in terms of response rate and progression-free survival. Given the fact that 40% of patients in the MIRASOL study had not been previously exposed to bevacizumab, do you believe the addition of bevacizumab to mirvetuximab may have the potential to further improve these outstanding results? And also, I have another question. Since patient selection is crucial for the success of this drug, at which point in time during the course of the disease do you foresee screening for folate receptor alpha?
Thank you for those questions, Dr. Colombo. I'm going to start with the easier one first, which is when do you screen? I do believe, and it will differ globally, assays differ and access to assays differ globally. So just speaking from a US perspective, where at the time of diagnosis now, or at least at the time of first recurrence, I would say most patients with ovarian cancer have undergone some sort of molecular profiling. And there's a number of commercial assays that are used. All of them give us kind of the same information regarding, fusions and mutations and amplifications. And we try to use that to tailor therapy in the recurrent setting for patients. I think this is going to be, and it's already actually been added to several of those platforms. So if you are sending one of those platforms, like the CARIS platform, for example, which provides immunohistochemistry, you're already getting PD-L1, status. For endometrial, you're already getting HER2 IHC. Now we're getting folate receptor alpha for ovarian cancer. So it's just getting added to those assays. And so I do believe that providers are going to know the folate receptor alpha status, not just positive, negative, but the percentage positive, because that will become important as new studies roll out very early on. And so you kind of plan for when you're going to bring mirvetuximab into that patient's kind of treatment discussion with that knowledge, from a pretty early time point. Right now, we just are seeing everybody kind of catch up because it's newly approved. And so, we're seeing a lot of patients asked to be screened who have had several lines of chemotherapy just because they've already been profiled and it wasn't available at that time. So right now we're seeing sort of a lot of catch-up sort of point of need testing where you test and then wait and see if you're positive and then you can use the medicine. But in the future, I think it's just going to be kind of like lung cancer. You just get everything about the tumor and you sort of know how to move forward. I think that's what we'll see with ovarian cancer in general moving forward, but certainly with folate receptor alpha. The question regarding bevacizumab is a great one. I'll remind the listeners that the approval for mirvetuximab, the accelerated approval, was based on the SORAYA study. That accelerated approval came in November of 22 based on SORAYA, which was a single-arm phase 3, which is another story in and of itself. A little over 100 patients, folate receptor alpha high, 1 to 3 priors, platinum resistant, and they had to have seen prior bevacizumab. Why is that? Well, for accelerated approval, what that means is that you're developing an area of high unmet need. And prior to this study, the only medicine that we really had that has ever shown a progression-free survival advantage, I'll remind you, Aurelia shows no OS advantage. So based on the progression-free survival advantage was Aurelia. So patients have that option, 1 to 2 priors, that was that clinical trial. And so after that is your high unmet need. And so that's where the SORAYA study was developed, so we could get that approved, and it was out to 3 lines of therapy, which is outside the Aurelia label. Now, MIRASOL was designed to be more inclusive. So you could have bevacizumab, but you didn't have to, because there's a lot of patients that can't get bevacizumab for a variety of reasons that we don't need to go back through. Sometimes they just choose to not get it, but they still benefit from this medicine. So that's why it was designed in this way. We did do an exploratory analysis that was presented at ASCO that shows the magnitude of benefit for patients who did and did not receive mirvetuximab was very consistent. So I would encourage you to look at that. However, we also have data from a pretty large, about 100 patients, a little less than that total, called Forward 2, and this is published, bevacizumab and mirvetuximab. In both platinum-resistant and platinum-sensitive, platinum-sensitive tumors that maybe had seen several lines of platinum or platinum allergic or whatever. And in both of those cohorts, the benefit of that combination was really high, almost 60% response rate in the platinum-resistant setting for medium and high expressors, and then almost 70% in the platinum-sensitive setting with very, very durable links of that response. And tolerability was just the 2 drugs. There wasn't any synergistic toxicity. So I do really like Mirv-Bev personally. Fortunately, it is NCCN listed. So in the United States, this is a US comment, it is NCCN listed for folate receptor alpha expressing tumors, not high. And I really appreciate that call out because Forward 2 was done in folate receptor alpha expressing tumors, and we see incredible benefit. And so it's allowed me to use Mirv-Bev in patients with 60% folate expression. And it works great. So I do think the addition of both is really smart. Patients don't lose their hair. It's every 3 weeks. The neuropathy is less. The hematologic toxicity is less. And so, to be perfectly frank, I feel very comfortable moving mirvetuximab up to my first platinum-resistant line of therapy with Bev and using that instead of, one of the Aurelia chemo partners. I know we don't have randomized data. I will acknowledge that, but I feel very comfortable that I'm doing the right thing by my patients with that combination. And so I think it's a great option for people. So more to come as we continue to evaluate the combination. It's in clinical trials in a study called Gloriosa for platinum-sensitive recurrent disease as the maintenance. So they get platinum-based therapy and then randomized to bevacizumab versus bevacizumab plus Mirv as a maintenance. And so we'll see more data from that as well.
Thank you for your answers, Dr. Moore. Now, I would like to move on to talk about some key updates presented in ovarian cancer, cervical cancer, and endometrial cancer at the ASCO meeting. And I would like to start with the ovarian cancer. The DUO-O trial was launched to investigate the safety and efficacy of maintenance therapy with bevacizumab monotherapy, 378 patients, versus bevacizumab in combination with durvalumab and olaparib, 378 patients, in patients with newly diagnosed advanced ovarian cancer and no BRCA mutations. A secondary analysis explored bevacizumab monotherapy versus bevacizumab plus durvalumab, 374 patients. All these patients received standard of care platinum-based chemotherapy, bevacizumab, durvalumab or placebo, and olaparib placebo as first-line treatment for ovarian cancer. The investigators found that the progression-free survival with the triplet maintenance therapy was significantly greater than with bevacizumab monotherapy. Because of the hierarchical testing, progression-free survival was tested first in the homologous recombination deficiency positive population, and then subsequently in the intention to treat population. And the median progression-free survival for the experimental arm in the HRD positive was 37.3 months versus 23 months in the control arm. And for the intention to treat population, the median PFS was 24.2 months versus 19.3 months. And both were highly statistically significant. So the effect was also consistent in all clinical subgroups, including surgical strategy, and particularly in patients who are HRD negative. In this population, the hazard ratio was 0.68, and of course, this was the most frequent patients in this trial. As in this population, the phase 3 PAOLA-1 trial did not show a benefit of adding PARP inhibition to bevacizumab, making DUO-O the first trial showing an effect of PARP inhibitors with ICI combination compared to an active control. The median progression-free survival in the intention to treat population was numerically greater with doublet treatment of bevacizumab plus durvalumab than with bevacizumab monotherapy, but this difference failed to reach statistical significance. So I think it's very important to underline that DUO-O is the first phase 3 clinical trial to meet its primary endpoint in ovarian cancer using an immuno-oncology agent, in this case durvalumab, in combination with a PARP inhibitor in this population. So my take-home message, although these results are very encouraging, we should remember that DUO-O lacked a control arm to isolate the effects of durvalumab by comparing the experimental triplet therapy to the olaparib bevacizumab combination that proved successful in PAOLA-1. But despite this limitation, this study opens the door to explore immunotherapy options in ovarian cancer management, which has thus far failed to yield a clinical benefit. The second study I would like to comment, which was presented at ASCO, is the abstract related to final overall survival results from KEYNOTE-826. This was a phase 3 study of pembrolizumab plus chemotherapy with or without bevacizumab versus placebo plus chemotherapy as first-line treatment for patients with persistent, recurrent, or metastatic cervical cancer. This double-blind phase 3 trial enrolled 617 patients with previously untreated, persistent, recurrent, or metastatic cervical cancer from 19 countries. The dual primary endpoints were overall survival and progression-free survival, the RECIST 1.1, assessed by investigator review, and each was tested sequentially in 3 different populations: the PD-L1 CPS 1 and higher, the all-comer, and the CPS 10 and higher populations. The trial met its primary endpoints at the first interim analysis, which we published in October 2021. At the 2023 ASCO Annual Meeting, we presented the results from the protocol-specific final analysis of KEYNOTE-826. The addition of pembrolizumab continue to demonstrate significant improvements in the overall survival and progression-free survival. The overall survival improvement was observed in patients with PD-L1, CPS1 and higher with a hazard ratio of 0.60, in the overall population with a hazard ratio of 0.63, and also in patients with a PD-L1 score 10 and higher with a hazard ratio of 0.58. All of these were highly statistically significant. So now, after a median follow-up of 39.1 months, the addition of pembrolizumab to chemotherapy with or without bevacizumab continued to show clinically meaningful improvements in overall survival and progression-free survival with no new safety signals in patients with previously untreated persistent, recurrent, or metastatic cervical cancer. Also, the objective response rates and the median duration of response were improved with pembrolizumab across these protocol-specified groups. Subgroup analysis indicated an efficacy benefit with pembrolizumab across most key subgroups, including, and this is very important, whether patients received concomitant bevacizumab or not. It is exciting that these results show a survival benefit of pembrolizumab in patients who are not eligible for bevacizumab, offering a therapeutic option in this population of patients with a high unmet need. Investigators concluded that these findings support use of pembrolizumab plus chemotherapy with or without bevacizumab as the new standard of care for patients with persistent, recurrent, or metastatic cervical cancer. We now move to endometrial cancer, and I'm going to talk about the ENGOT EN6 NSGO GOG 3031 RUBY trial, which is a phase 3 study of dostarlimab or placebo plus chemotherapy, followed by dostarlimab placebo maintenance in primary, advanced, or recurrent endometrial cancer. The primary endpoints, progression-free survival and overall survival, were already published on the New England Journal of Medicine, but there were two abstracts presented at ASCO 2023 regarding RUBY trial. The first was related to the assessment of RUBY trial outcomes by blinded independent central review. This analysis showed that the hazard ratios for progression-free survival mirror those per investigator assessment and confirm the benefits of adding dostarlimab to chemotherapy for women with primary, advanced, or recurrent endometrial cancer with mismatch of repair deficient, microsatellite instability, high tumors, and also in the overall study population. The other interesting abstracts regarding RUBY trial refers to the quality of life assessment among the 494 patients enrolled in the study. The EORTC QLQ-C30 and EN24 were pre-specified secondary endpoints. And the investigators reported the assessment for cycle 7 at the end of chemo and cycle 13 and at the end of 1 year of study. So if you look at the pro outcomes, they were similar for dostarlimab plus chemo and placebo plus chemo through the chemo period. And further, no differences across the 3-year period between the two arms were reported. However, the mean change from baseline to end of treatment showed actually an improvement in back pelvic pain for dostarlimab plus chemotherapy, and on the other hand, a deterioration in the global quality of life, social functioning, body image, and also change in taste for patients on the placebo plus chemotherapy arm. So in conclusion, dostarlimab plus chemotherapy significantly improved progression-free survival while maintaining health-related quality of life, further supporting its use as standard of care in patients with recurrent and advanced endometrial cancer.
Professor Colombo, thank you so much for those great summaries of these very important abstracts. ASCO 2023 was very exciting for gynecologic cancers and lots to unpack. I have a couple of questions for you on these studies. So one of my kind of key questions is for KEYNOTE-826, which is a practice-changing clinical trial improving outcomes for women with cervical cancer. As you know, once 826 was initially presented, despite it not being an analytic endpoint, the regulatory authorities excluded patients, or tumors, I should say, excluded tumors that were PD-L1 negative from authorization to use pembrolizumab with chemotherapy and bevacizumab in the metastatic recurrent setting based on A, a particularly small number of patients, and B, the subset analyses that showed that they were kind of right at one, so didn't benefit. And I think we all know that subset analyses are non-analytic, so that was somewhat surprising. I was really interesting when I saw Dr. Monk present 826, and he put that subgroup slide up, and it looks like it's gotten better. The updated data seems to suggest that those tumors with PD-L1 negative status actually do benefit from pembrolizumab. And again, I just said subsets are hard to analyze, and I'm going to overanalyze this one. But how do you comment on this? Do you think it could impact current regulatory restrictions for this kind of high unmet need subset, or do you think it's real? Like, what is your take on that particular subgroup?
I think affordability and drug access is one of the most important issues when it comes to regions with the highest incidence of these cancers. Actually, I recently spoke with some of these drug companies in this respect, and they reassure me that they're putting in place several actions to help, such as, for instance, individual-based early access program or early access to clinical trials. However, I think this will remain a major problem in developing countries. As far as the PD-L1, the hazard ratio for negative PD-L1 group in the original paper was 1.0, and in this presentation is 0.87. Actually, there are still 7 PD-L1 negative patients who are on study, and 6 of them are in the pembro arm and only 1 in the placebo, which is, of course, very encouraging. So not sure if this will be enough for the regulators to change the label. I must say that only 69 patients in the KEYNOTE-826 had a CPS score of 1 and less than 1. So for me, it is almost impossible to draw any definitive conclusion with this very, very low number.
I want to move on to your second abstract, which was the DUO study. Exciting. We've been talking about the 4 randomized phase 3 clinical trials in ovarian cancer that are including immune checkpoint inhibitors with PARP inhibitors, some with bevacizumab. They're all different studies, but kind of similar. We've been waiting for them, and here's the first one to report out its high-level results at the time of interim analysis. So very exciting. Sometimes it's hard to be first, though, so we're going to be overly critical. So my question for you really is, how is DUO not just PAOLA-1 with durvalumab? How do you address the criticism that I can't tell the value of durvalumab over the value of just using a PARP inhibitor with bevacizumab, just like we did in PAOLA-1? So I'd like that sort of my question A for DUO. And my question B is, this was really a study of immune checkpoint inhibitor, not of PARP inhibitor, and yet PD-L1 was not studied as a subgroup, as a primary endpoint. And so I was curious as to why you think that is, and whether or not it strengthens versus weakens the study, not having that kind of called out PD-L1 group, positive group, identified and evaluated. So I'd appreciate your responses.
You're right. DUO is missing an important control arm, which is the olaparib and bevacizumab arm. When it was designed, the results of PAOLA-1 were not available. And when they became available, it was too late to change the study design because too many patients were already included. I recognized that it's now very difficult to identify the real contribution of durvalumab to this combination and which drug is the main driver for the superiority of the triplet compared to the standard arm. It is, however, intriguing to see has improved progression-free survival in the HRD negative population, since this was not seen in the PAOLA-1 study, and therefore I think represents a new interesting finding. As far as PD-L1 was actually studied, and in the subgroup forest plot, you can't see any difference in progression-free survival according to the PD-L1 status, which was tested according to TAP5, high and low.
I want to thank Professor Colombo for this great discussion and analysis of key updates and new data presented at ASCO 2023. I hope our listeners have found the discussion educational and that can inform their clinical practice. And this concludes our segment on key studies at the ASCO 2023 Annual Meeting. Thank you so much for joining us and have a great day. Goodbye.
Thank you very much, Dr. Moore and Dr. Colombo, and thanks to you, the listeners, for joining us. As a reminder, to view the full program, key findings and expert recommendations in gynecologic malignancies, independent 2023 Conference Congress coverage, and to download a highlights slide set including the various studies associated with this discussion, please click on the link in the show notes. As always, thanks for listening.